All right, we are here to talk about the citric acid cycle, which you may have heard. They're kind of getting away from this, so you may not have heard it called this, but it used to be called the Krebs cycle because it was discovered by Hans Krebs a long time ago. I don't know, 50s, 40s, 30s, 60s, somewhere, probably getting close to 75, 100 years ago. Anyway, the citric acid cycle, your three I can statements are: I can describe the process of the citric acid cycle, I can list the reactants of the citric acid cycle, and I can list the products. So reactants, products, and process. So if you'll remember, pyruvate oxidation, we finished up with 2-acetyl-CoA. Okay, so that's where we're picking up. This is just like a story that keeps going. So we're going to start with an acetyl-CoA, and there were two of them, because if you remember, glucose split into two pyruvates. That's fine. There we go. And then those pyruvates lost a carbon, so now we have 2 acetyl-CoAs. So each of these acetyl-CoAs is going to go into this, but I'm going to start by showing off just one. All right, so what happens is acetyl-CoA comes in and it binds with a 4-carbon molecule. Well, 4 plus 2 is 6. This molecule, and it's the only one you need to know its name, is citric acid. Most cycles that have a name like this are named after that first molecule. So the first molecule is citric acid. That's why it's called the citric acid cycle. Then this citric acid, this 6-carbon molecule, loses a carbon. It's going to leave in the form of CO2. So what happens is we have some atmospheric oxygen come in, pick up the carbon, and leave. Okay? Because we broke a bond, so we had 6 carbons. We broke one off, we're down to 5. Because we broke a bond, we released energy in the form of electrons, and that goes to NAD plus to make NADH. So those electrons moved from the citric acid to the NAD plus. So this was reduced because it gained electrons, and this guy up here was oxidized because it lost electrons. Okay? So in that step, we made a CO2 and we made an NADH. That leaves us with a 5-carbon because we had 6, we lost 1, we're down to 5. Okay? Then we're going to lose another one. So we have another CO2 coming out. And again, that's an atmospheric oxygen coming in, picking up that CO2 and leaving. So this is an aerobic cycle. Okay? There's actually 3 things I want to add up there in a minute. Because we had a 5-carbon and we lost a carbon, we're down to 4. And when we broke that bond, when we broke that 4th carbon off to give us a 4-carbon, that bond released electrons, energy in the form of electrons. And so we know that those electrons moved from this 5-carbon to NAD+. So that means NAD+ was reduced because it gained electrons, and this guy up here was oxidized because it lost electrons. This is a fairly high energy molecule, which just means that bonds in it were high energy, so it's high energy bonds. And we're going to rearrange it, take out some of that energy to make another 4-carbon molecule that's low energy, and that's going to be the 4-carbon molecule we started with, and so that's going to start our cycle all back over. All right, so when we break those bonds, we're going to release energy in the form of electrons, and some of that is going to go to NAD plus to make an NADH. Okay, some of that is going to go to a new molecule, which is NAD plus's cousin, FAD++, or sometimes you'll see FAD2+, and it's going to pick up even more electrons. So it has 2 positives. It picks up 2 hydrogens and 4 electrons. Okay, so the 2 positives, 2 more positives, 4 electrons, and that gives us FADH2. Okay, not real important, but in case you're wondering, that's what's going on. All right, so this guy picked up electrons, so it was reduced. I'm just going to put RED. This one picked up electrons, so it was reduced. All of those electrons came from this one, so this one was oxidized. And we also were able to harvest a little bit of that energy directly into ATP. And so we have an ADP being converted to an ATP, and that is through substrate level phosphorylation. Okay, so an enzyme just comes in, uses that energy that was released from this molecule to put that phosphate on ADP to make ATP. The next thing, or one of the things I want you to know, is where this happens. It happens in the matrix of the mitochondrion. So if you look at a mitochondria, it kind of looks like a peanut, and it has that real wiggly inner membrane. The matrix is the very center, and it's kind of a jelly-like fluid or substance in there. I just realized my cycle looks a little wonky, but pretend like that's a good circle. All right, so let's look at products and reactants. All right, reactants, what went in? Okay, and and one other thing I need to mention is that all of this, not making a good line there, all of this happens times 2, because if you'll remember, glucose, 6 carbon, broke into 2 pyruvates, 3 carbons. Each of those lost a carbon, so we have 2 acetyl-CoAs, and this is only showing 1. So this is happening twice for every glucose. The reactants, I'm actually going to make 2 columns. So I'm going to make per acetyl-CoA, and then per glucose. And you could get asked this question either way on the AP exam, so make sure you read the question really carefully. Are they asking per turn of the cycle, so 1 turn of the cycle, 1 acetyl-CoA, or are they asking per glucose, which means we had 2 turns of the cycle? All right, so let's start with per acetyl-CoA, because then all we have to do is multiply it times 2 for that. Reactants. What went in? We had an acetyl-CoA go in. We had 2 oxygens coming in. Whoops, 2 O2. Okay, we had 1, 2, 3 NAD pluses coming in. We had an FAD++ come in. We had an ADP come in. Okay, and I think that's it. We're not going to count this as a reactant because it rides through the cycle over and over again. Okay, so that means per glucose we have 2 acetyl-CoA, 4 oxygen, 6 NAD+, 2 FAD++, and 2 that should be a DP. All right, so let's look at products. Let's see if I can squeeze these over here, because I'm going to do the same thing. I'm going to make 2 columns. So per acetyl-CoA, and then per glucose, because I want you to see both just in case you have to deal with both. So the first thing I'm going to notice that comes off is a CO2, and there are 2 of those. So I have 2 CO2s coming off for every turn of the cycle. I have 1, 2, 3 NADHs, 1 FADH2, and 1 ATP. I'm getting ahead of myself. Okay, and so per glucose, we're just going to double that. We have 4 CO2, 6 NADHs, 2 FADH2, and 2 ATP. Okay, and those ATPs again are formed by substrate level phosphorylation. So I can describe the process, I can list the reactants, and I can list the products. And so that is the citric acid cycle. Hope that helps. If you have any questions, if you just want to go over it again, let me know. Like I've said with the other ones, drawing it really helps. Paying attention to this breakdown every time you lose, you break something off, you make an NADH. That helps you remember that kind of stuff. So like I said, I hope that helps. I know it's a lot of details, but honestly, there's a lot more going on that we're not showing. But if you have questions, just let me know.